Okay. First, a couple of points I want to make. Don't forget that today you're doing quiz 7. And you have all day to do that. Some of you have taken the quiz already. You have until midnight to finish the quiz. You have 2 attempts. The other point I want to make is that next week is vacation. It's Thanksgiving week, so we will not have class on either November 22nd or the 24th. We'll meet again on Monday, November 29. So don't go to class next week. You see the first slide, overview. Tell me.
Yes, we can.
You can, okay. So we'll proceed. I want to get through this in 60 minutes because you have 15 minutes out of your day for the quiz. So this is to remind you where we've been and where we're going. The 4 topics: energy, enzymes, photosynthesis, and respiration. We've done the first 3. Talked about energy, what it is, how life forms use it. We've talked about enzymes, how they work. Photosynthesis is the capture of energy from sunlight. And today we're going to talk about respiration, derivation of energy from food. So once photosynthesis has provided food, how do we use it? So there are 2 ways. Aerobic respiration and fermentation. And here I have a contrast, an overview, contrasting the 2 types, aerobic oxidation and anaerobic oxidation. In aerobic oxidation, that's what we do all the time when we breathe. It's also known as cellular respiration. A key in this process is glycolysis. Glycolysis is literally lysis of glucose, which means breaking of glucose apart. And that leads to what is called the Krebs cycle. And we'll talk in detail about that. The Krebs cycle involves electron transport, and that leads to oxidative phosphorylation. We get a lot of energy from that. Now, supposing there's no air, it's anaerobic, and the condition then is known as fermentation. And notice that again, it leads to glycolysis. So you end up with glucose, end up breaking it. But at this point, the next step, Krebs cycle, does not happen. So in the case of, say, your muscles, with the absence of oxygen, you get lactic acid. If, on the other hand, it's a microbe, for example, a yeast, you get alcohol and carbon dioxide. And both of these products are very important to us humans. So let me go back to the structure of a cell to let you know where the action is. You see, and this is a plant cell, and so it has chloroplasts. And we've been talking in the last period about chloroplasts and what goes on in them. Well, animal cells don't have chloroplasts, but they do have mitochondria. And by the way, plant cells also have mitochondria, so don't forget that. Plant cells respire just like animal cells do, plus they have chloroplasts. Animal cells have only mitochondria and only respiration. So here's an overview of what we're going to talk about today. We're going to start with glycolysis, which is the conversion of glucose to a substance called pyruvic acid, or pyruvate for short. And when this happens, you get a substrate level phosphorylation, 2 ATP molecules. And this occurs in the cytoplasm. Cytosol is cytoplasm. The pyruvate is capable of crossing the mitochondrial membrane into the mitochondrial space, and I'll show you a close-up of this in a minute. And then it enters the Krebs cycle. Well, the Krebs cycle also has a substrate level phosphorylation, 2 ATP molecules, but it generates, both of these first two steps generate electrons that are transported by these electron carriers, NADH and FADH2. These carriers enter the electron transport chain and result in oxidative phosphorylation. The final step of that is chemiosmosis, and that is where we get most of our energy. Most of our ATP comes from oxidative phosphorylation, and we can count them up a little later. But first, let's take a closer look at glycolysis. So you start with a molecule of glucose. It's usually a cyclic molecule. It can be linear. When glycolysis occurs, two things happen. One, 2 molecules of ADP are converted to 2 molecules of ATP. And at the same time, 2 molecules of NADH are generated. And you get these 2 pyruvate molecules. So we go from a 6-carbon compound to 2 3-carbon compounds. Now, this shows glycolysis happening in one step. It's actually 9 steps shown here, starting with glucose. You don't need to know all these intermediates. What you do need to know is that glycolysis consumes 2 molecules of ATP. It generates 4 molecules, so there's a net ATP generation in glycolysis of 2 ATPs. For 1 glucose, there's a net of 2 ATPs.
I have a quick question.
Yeah, hi.
Is glycolysis going to be on the test today? Or a quiz, whatever it is?
No.
Okay. What will be on the quiz today?
The quiz covers molecular genetics, enzymes, energy, and photosynthesis.
Oh, okay. Thank you.
Uh-huh. Well, this slide is entitled The Grooming of Pyruvic Acid. The summary of the information here is in the upper right. 1 glucose, which has 6 carbons, is converted to 2 pyruvates, each of which has 3 carbons, and those go to 2 acetyl-CoA molecules, which are 2 carbons. Where did the other carbons go? To carbon dioxide. So when we breathe in oxygen, we exhale carbon dioxide. So this is a close-up of what happens first in the cytoplasm. We have a pyruvate molecule, 3-carbon compound. When it enters the mitochondrion, so it's produced in the cytoplasm. It enters the mitochondrion across the membrane, and when it does, it generates a molecule of carbon dioxide and NADH. And because it loses carbon dioxide, the product is acetyl-CoA, which is a 2-carbon compound. And this is the first step in the Krebs cycle. You've seen this before. When I told you about ways to produce ATP, this is the first way I described. It's called substrate level phosphorylation. And that's what I've just described here. Substrate level, where you generate 2 ATPs from 1 molecule of glucose. That is substrate level phosphorylation, where you take ADP plus phosphoenolpyruvate with an enzyme called pyruvate kinase that's converted to pyruvate plus ATP. And the ratio here is 1:1. So if you start with 1 glucose, you get 2 PEPs plus 2 ADPs generates 2 pyruvates and 2 ATPs. So there's no amplification of ATP production in substrate level phosphorylation. And this shows the same thing in a more graphic form. The substrate PEP, which has this phosphate group on it, phosphoenolpyruvate, and it transfers this phosphate group to ADP, and the product is ATP, adenosine triphosphate, plus pyruvate, which is no longer phosphorylated. This is the third way I told you of generating ATP, and it's called oxidative phosphorylation. We've already gone over way 2, which is photophosphorylation. That was last time. But now we're going to focus on oxidative phosphorylation, because that's where we get most of our energy, most of our ATP. So let's just look at the diagram, starting at the top with glycolysis. In glycolysis, glucose is cleaved to produce 2 3-carbon compounds, pyruvic acid, generating 2 ATPs, substrate level phosphorylation, and 2 NADH. And so these go off and will accumulate. The pyruvic acid is converted to 2 molecules of acetyl-CoA. This is an important molecule because it's the first step in the Krebs cycle. And this conversion results in 2 more NADHs and 2 molecules of carbon dioxide. Then the Krebs cycle is activated and it turns. I'll show you the steps in this cycle. And as it does, it generates a lot more NADH and another electron carrier called FADH2. And another consequence of the Krebs cycle is that more carbon dioxide is generated so that so that from 1 molecule of glucose, we end up with 6 molecules of carbon dioxide. And there's another, as this cycle turns, there's another substrate level phosphorylation generating 2 ATPs. So that's really the beginning and the end of what happens in the Krebs cycle. It generates a little bit of ATP and a lot of electron carriers, reduced electron carriers. These enter the electron transport chain. And they're split, the electrons go through the membrane, and the protons accumulate on one side of the membrane. This is called oxidative phosphorylation. And as the electrons move down this chain, going from one carrier to another, to another, to another, they end up, the last electron acceptor is oxygen. That's why we need oxygen. And because it accepts electrons, it's converted to water. So we end up with, as I said earlier, 6 carbon dioxides and 6 molecules of water from 1 molecule of glucose. Plus, because of oxidative phosphorylation and chemiosmosis, 34 molecules of ATP. So the total is 38. So from 1 molecule of glucose going through oxidative phosphorylation, we end up with 38 molecules of ATP, whereas in substrate level phosphorylation, the total is 2. So this is really crucial, really important, because this is where we get most of the energy that we use. And I've got a diagram here that you've seen before, which illustrates chemiosmosis. So at the end of electron transport, protons, the electrons are in the membrane, moving along, losing energy, and they're generating protons on one side of the membrane. So there's a proton gradient. You saw this in the video that I showed you earlier. So this high concentration of protons will move across the membrane through a complex called ATP synthase. And that converts ADP to ATP. So that's where we get these 34 of the 38 molecules of ATP. Make sure you remember what chemiosmosis is. And I just wanted to remind you, when we go back here, during oxidative phosphorylation, as electrons move through the membrane along the electron transport chain, what's really happening is a series of these handoffs where electrons are handed off from a substrate to a carrier, and this carrier hands off to another carrier, and they keep going from one carrier to the next until to the end, till they finally hand off to water, or to oxygen to make water. So that's why this I've shown you before, but the reason I am is to remind you that these handoffs is what's going on in oxidative phosphorylation. Now this shows it more graphically with a membrane, with these proteins that are embedded in the membrane. And these contain the carrier molecules, the electron carriers. So NADH is converted to NAD from the Krebs cycle, And that provides electrons for a flavin, for coenzyme Q, cytochrome B and C, cytochrome A and A3, and finally, electrons are handed off to oxygen to make water, the final electron acceptor. And now in the process of doing all this, every time there's a handoff, more protons are generated. The protons keep accumulating on one side of the membrane so that there's a gradient across the membrane. And when they move through the membrane, through the ATPase complex, ATP synthetase complex, this is called, this large blue thing is called chemiosmosis. And that's what actually generates the ATPs. So electrons moving through the membrane, protons accumulating on one side of it, they provide the energy to synthesize ATP from ADP. Just to remind you or make clear to you where all this is going on, remember I showed you a picture at the beginning of a plant cell that had mitochondria and chloroplasts. We're not talking about chloroplasts now, only mitochondria. So if you take a single mitochondrion and look inside it, what you see is a series of highly folded membranes called cristae. And there are two critical spaces here, this intermembrane space between the cristae and the outer membrane, and the membrane, well, the matrix. So the intermembrane space and the matrix are crucial because as oxidative phosphorylation occurs and electrons are transported within the membrane, the protons are accumulated in the matrix. And when they are transported across into the intermembrane space, that's when ATP is synthesized. So this inner mitochondrial membrane is analogous to a thylakoid. It's not the same thing because it's in a mitochondrion instead of in a chloroplast, but the same series of events happens. That is, electrons in both thylakoids and mitochondrial cristae, electrons are transported, generating a proton gradient, which generates ATP. So that is important to remember, that this ability to make ATP goes on in both plants and animals. In animals, it's in the mitochondria. In plants, it's in both. So let's take a closer look then at the Krebs cycle, also known as the citric acid cycle or the TCA cycle. The first step in it, as I said earlier, is acetyl-CoA, a 2-carbon compound. So that, along with oxaloacetate, makes citrate, a 6-carbon sugar, or not sugar, compound. And then the cycle turns. It goes from one intermediate to another, to another, to another, and completes the cycle with oxaloacetate. And what happens here, there are 2 points at which the carbon chain is reduced with the yield of carbon dioxide after isocitrate and after alpha-ketoglutarate. And at several points, reducing power is generated in the form of NADH or FADH2. And I'll remind you that during one turn of the Krebs cycle, there is one substrate level phosphorylation resulting in ATP synthesis. Well, this is an important picture, and there are several points I want to make here. First is you look at a cell. Here's the nucleus, here are the mitochondria. A blow up of a mitochondrion, and then we peer into the mitochondrion itself. When the mitochondrion is sitting in the cytoplasm, there's glucose in the cytoplasm from food you've eaten, and that's converted to pyruvate, which crosses the membrane. We have pyruvate inside. That's converted to acetyl-CoA, which is the first step in the Krebs cycle, the TCA cycle. So the Krebs cycle turns, it does all the things I've just been telling you. It makes CO2, makes a little bit of ATP, and a lot of electron carriers. These electron carriers then enter the membrane and go through a series of steps where they're losing energy. And as they do, they generate protons. And you can see that the protons, all these H pluses, are accumulating in this intermembrane space. This is called a Christie. And finally, when the electrons get to the end of the chain, they're accepted by each one by a half molecule of oxygen to make water. This proton gradient that's generated, you have this high concentration of protons inside, they can now diffuse through the membrane, through these ATP synthase complexes shown here in purple. So these are the sites of chemiosmosis, 3 shown here. And this is where large amounts of ATP are synthesized. So this is starting with sugar and ending up with ATP. There's another way to do it, and that is to start with fat, shown here in the upper right. Fat is triglycerides. They can be converted to fatty acids. And then there's a process known as beta oxidation, which converts fatty acids to acetyl-CoA. That is the first step in the Krebs cycle. And from there on, it's the same. You end up with ATP being generated along with carbon dioxide and water. So think about when you eat. If you just ate a handful of candy corn, what would go on in this picture? Candy corn is pure sugar.
I mean, that would probably turn into glucose that then starts the process that goes into making it into what acetyl CoA, which is the first step of the Krebs cycle, and then it just kind of goes through everything else.
Yeah.
Also, I got another question. I know you kind of just said this, but so the protons they're in the inner membrane space, not the inner membrane or the matrix.
Right.
And maybe I missed this, but how exactly do they get in there in the first place? Is that just, are they always in there, or?
They get in there from starting with glucose, you end up with pyruvate, and then the Krebs cycle generates these intermediate carriers.
Yeah.
And let's go back to what the carriers do. So remember, each step here is a redox reaction.
Okay.
So you end up with a reduced carrier and an oxidized carrier. And when that happens, a proton, see this little red
Yeah,
H here? That's a proton.
got you. So when it
So-
goes through the reduction, it generates a proton essentially.
Yeah,
Okay,
so the electrons keep getting passed back and forth and carried along. And at each step in this redox reaction, a proton is generated.
okay, gotcha.
So, let's see. Yeah, so that's what's happening in the Krebs cycle. You're getting these reduced carriers that then enter the electron transport chain, and the electrons go through the membrane. The protons get they become accumulated on one side of the membrane in this intermembrane space. So go back here, see this intermembrane space? That's where the protons are accumulated. And then when they go through the membrane, they end up in the matrix, and that's what generates, that's chemiosmosis. That's these purple bodies that generates the ATP.
Okay, gotcha. Yeah, I think the picture on right now explains it, or it makes it a lot easier for me to visualize, because I was really confused about the intermembrane and the intermembrane space, all the different membrane spaces.
You're right. Yeah, the two critical ones are the intermembrane space, which is a space between a crista and the outer membrane, and then the matrix, which, well, the matrix you can see here, the gray part, this mitochondrion is the matrix, and the inner membrane, you can see in the intermembrane space is where the protons accumulate.
Yeah, that makes sense.
Okay, so you just told me what would happen if you ate a handful of sugar. What if you had a salami sandwich with cheese? What would happen? Cheese is pure fat. Salami is mostly fat. Any thought? You
I have
certainly,
a pretty good guess. I don't know if anyone else wants to answer that though.
oh, go ahead.
Yeah, well, it goes down, so it's a triglyceride, and that turns into the fatty acid, and then it goes through the beta oxidation process, which then turns it into the acetyl CoA. Then it starts the Krebs cycle again, and it's the same thing we've all been talking about, or
Right.
you've been talking about.
So depending on what your diet is, determines which way this pathway is going to run. It'll run from left to right if you're eating primarily sugars. It'll run from right to left if you're eating fats.
If you ate something that was fatty and sugary, does it do the same process at just both ways, or does it have to be one of the other ways?
No, most of our diet is a mixture.
Yeah.
So the fact is then that both pathways would be running.
Okay,
But if
gotcha.
you did something extreme, like eat pure sugar, then only one would run. Or pure fat, like a hunk of cheese, only the other one would go.
Yeah, probably wouldn't feel good either.
You may not. Okay. Let's see how we. I've been talking about acetyl-CoA. It's called a crossroads compound. So here's a little more about acetyl-CoA. It's the beginning of the Krebs cycle. So it's a high energy fuel molecule. It's a common pathway point for the extraction of energy from carbohydrates, fats, and proteins. So that's shown here as this acetyl-CoA is this high energy molecule, and it can get its energy from either sugars or fats. And in that sense, it's catabolic. So these pathways are catabolic. They're tearing molecules apart and releasing energy. Acetyl-CoA can also be a building block for fats. So if it's used in that way, it's anabolic. It requires energy to go from individual molecules to polymers.
I don't know if you're gonna cover this, but why would you need it to turn into a fat? Or why does that process happen?
Fat is a storage substance. And so if you have too much of it, then you would reverse this pathway and convert acetyl-CoA to fat.
Oh, okay, because then it stores it in your body.
Yeah. The same way that plants convert small molecules to complex carbohydrates. Okay, so acetyl-CoA can be either catabolic or anabolic. And this is a structure of it. The right-hand side is ADP. See, it has 2 phosphates here. Then if you add pantothenic acid, which is a vitamin, you end up with coenzyme A. And then if you add an acetyl group to that, it's acetyl-CoA. So this recapitulates what I've been saying. You eat food. Here's a pizza. It's got all kinds of high energy molecules in it, largely fats, complex carbohydrates, and proteins. And the important point about this summary is that all the energy from all types of food ends up in the Krebs cycle. And that feeds the electron transport chain, which leads to phosphorylation, ATP. This shows the same thing slightly differently. The major foods that we eat: proteins, polysaccharides, and lipids. Nucleic acids aren't really an energy source for us. So these all end up going to acetyl-CoA, which is the first step in the Krebs cycle. Another way of, it's not another way, it's a reiteration of what we've just been talking about, starting with glucose, ending up with chemiosmosis here in the membrane as a result of all these steps we've been talking about. Now, what happens if you don't have oxygen? Fermentation. So here we have all the same steps, but if there's no oxygen, what happens? Remember, oxygen is the final electron acceptor. So this slide is different from previously. You see that oxygen is crossed out. There is none. So if there's no oxygen, what happens? Electron transport chain stops. It can't deliver electrons to oxygen to make water. So it stops, and that backs up. The Krebs cycle stops, and you end up with a block right here, the red bar, a block with pyruvate, which is then diverted to something else. But it does not go into the Krebs cycle. It does not yield high amounts of ATP. So that leads me to a discussion of fermentation. One type of fermentation results in ethanol. And this happens, so the products of fermentation depend on who's doing it. If it's yeast, you get ethanol. So you have glucose here, glycolysis, you get 2 substrate level ATPs produced, and it makes pyruvate. And this is all going on in the cytoplasm. Pyruvate is converted to acetaldehyde with the production of 2 molecules of carbon dioxide. Then this acetaldehyde is reduced because glycolysis is generating reducing power, NADH. So acetaldehyde takes advantage of these hydrogens to be reduced to ethanol. Ethanol is a 2-carbon alcohol. And this could be wine. It could be used for wine. So this is what happens if yeast is involved and there's no oxygen. What if the muscle cells have no oxygen? What happens? Well, you get the same first step. Glucose undergoes glycolysis, and you get pyruvate molecules, but no carbon dioxide is generated. Instead, the pyruvate is directly reduced to 2 molecules of lactate, lactic acid. So you have pyruvate, a 3-carbon compound. Lactate is a 3-carbon compound, which is just reduced pyruvate. So without oxygen, lactate accumulates. And this is relevant to all of us when we get tired. You've all done vigorous exercise and your muscles burn, and here's why they do. You have glucose in the muscle being converted to lactate, which accumulates, and the lactate then is metabolized back to glucose. So why do your muscles burn? Because you have inadequate oxygen. Your muscles need ATP. Where are they going to get it? They could get it either from substrate level phosphorylation and have it immediately, or they could wait for oxidative phosphorylation, which would take a while, and get a lot of ATP, but they don't have time to wait. So if we go back, under anaerobic conditions, a little bit of ATP is generated. And so if the muscles really need it, they don't have any oxygen, they're going to keep doing this to get more ATP, but every time they do, they make more lactic acid. And so you feel this burning sensation. And that's really all I want to say about it. Lactic acid builds up because there's no oxygen, and you have to stop and rest so that you can catch your breath, get some oxygen into your muscles, and get back to oxidative phosphorylation.
Professor, I have one question.
Yeah.
So this is really something I've been hearing a lot because I'm a cross country runner, and we have this type of workout that is called the lactic threshold. And what we basically are training is to like avoid this kind of muscle thing that's going on. Is there a way that you can probably reduce how much lactate is being generated on your muscles? Is it just by oxygen? Is it the only way?
You're running cross country, yeah. So you're putting a lot of stress on your muscles, and they're getting low on oxygen. And I don't know I don't know, I'm not an exercise physiologist, so I don't know if there's a way that you could reduce the amount of lactate that accumulates.
I have a potential answer.
Uh-huh.
So I was gonna say something similar, but I do breath work and I teach breath work to people. And what you could do is you could, before you go on a run, you could do something like Tumo or like Wim Hof breath work, where you like front load all the oxygen, and you probably have exercises your teacher might show you for that. But you could probably do something like that, where you do a breath work that then builds more oxygen. So you have more in your reserve. So as you're running, it won't be as bad, but I don't know that for sure. That's just kind of like just a thought that I've had.
Well, it makes sense that if you could say super saturate your body, your lungs, your muscles with oxygen, then lactic acid would accumulate more slowly, or at least later. So if you're in a cross-country run, you might be able to run a few miles farther without exhausting your muscles.
Right, so this is why people say that altitude is like it's harder to run, because it's like you build more lactate because of less oxygen.
That's right.
Interesting. Okay.
So well, my question is, is there a threshold of the amount of oxygen that you can actually retain in your body? Because because it sounds like you're trying to, like you would carbo load, you would try to oxygen load, and it seems like there would be a threshold by which that your body would not take in any more oxygen.
Yeah, there's got to be a lid on it. But I think this has to do with conditioning. So the more you condition your body to absorb oxygen, the better shape you're in to avoid fermentation. Remember, if you have oxygen, you're going to go back to this. If you have oxygen, so this is not blocked, then you're going to go back to oxidative phosphorylation, which makes a lot of ATP, and that's music to the ears of your muscles. So if you front load a lot of oxygen, you're going to provide opportunity for oxidative phosphorylation, and you'll delay this. Excuse me, delay this, delay the accumulation of lactate. Okay. So this slide is just a summary comparing sites of fermentation, whether it's microbes or muscles. And in the case of microbes, yeast in particular, what you get from fermentation is two products that are important to us: carbon dioxide and ethanol. So some of you may be cooking for Thanksgiving next week, and you may bake bread. So how are carbon dioxide and ethanol important in bread making? Any of you have baked bread? What are the ingredients?
The yeast that makes it.
Yep.
Yeah.
Yeast, right. And so why do you add yeast?
So the bread rises.
Right, why does it rise?
Fermentation that makes the yeast.
Yeah, it's fermentation.
Carbon dioxide.
Carbon dioxide, it's a gas. Right, so you add yeast. The conditions are anaerobic in your yeast, in your bread dough. And so it makes carbon dioxide. It also makes ethanol. What happens to it? Where do you bake the bread? In
The
your
oven.
in your
So
oven.
it evaporates, correct?
It rises.
Yeah, it evaporates. Because you are baking the bread in the oven, and the carbon dioxide remains trapped as the bread bakes, but the ethanol escapes because it's volatile. Okay. So this is a gross overview of cellular respiration. Starts with glucose, needs oxygen. That's converted to carbon dioxide and water, and you get, for every molecule of glucose, 38 ATP molecules. Overview of where it all happens. In the cytoplasm, glycolysis occurs. Within the mitochondrion, the Krebs cycle occurs, and electron transport, chemiosmosis, and phosphorylation. High levels of phosphorylation occur in the mitochondria. Glycolysis, glucose, the 6-carbon sugar, it results in 2 molecules of 3-carbon sugar, pyruvate, generates 2 ATPs and 2 NADHs. I won't go over this again. It simply shows the membrane with the electrons being transported within the membrane. With each transport, remember, each of these is a redox reaction. The electrons are losing energy, and where does that energy go? It goes to the transport of protons to one side of the membrane, so that a proton gradient accumulates, high concentration of protons, which then pass through this complex ATP synthase to make ATP. Now, I want to have just enough time left to make 2 comparisons. First is cycles, to compare the Calvin cycle, which remember we talked about in photosynthesis, and the Krebs cycle, which we talked about today for respiration. So the definition, Krebs cycle is part of the respiration process. Calvin cycle is part of photosynthesis, occurs in the chloroplast. The location, mitochondria versus chloroplasts, the process is catabolic. In the case of the Krebs cycle, that is, it's tearing complex molecules apart and releasing energy. The Calvin cycle is anabolic. It's using energy to build up complex molecules. In the case of the Krebs cycle, it produces ATP and carbon dioxide, so they're produced in the Krebs cycle. In the Calvin cycle, they're used. ATP and carbon dioxide are used to make sugars. The Krebs cycle occurs in all aerobic organisms, both plants and animals, whereas the Calvin cycle only occurs in plants because it occurs in chloroplasts. The Krebs cycle oxidizes glucose. That means it breaks it down. The Calvin cycle produces glucose. And finally, the Krebs cycle it occurs only in the presence of oxygen, whereas the Calvin cycle is indifferent. It doesn't matter if oxygen is there or not. So this is just, I think, a convenient comparison of the 2 cycles, and you will need to know the distinction between the 2 kinds of cycles for the upcoming tests. And finally, another comparison, in this case, between oxidative phosphorylation and photophosphorylation. And not included here is substrate level phosphorylation, which is a different process altogether. So oxidative phosphorylation produces ATP using enzymes, a whole series of enzymes, and oxygen. Whereas photophosphorylation ATP is produced using sunlight as a source of energy. The energy source for oxidative phosphorylation is molecular oxygen and glucose, whereas the energy source for photophosphorylation is sunlight alone. Location, mitochondria versus chloroplast. Occurrence, oxidative phosphorylation occurs during cellular respiration, whereas photophosphorylation occurs during photosynthesis, that is only in the light. And the final electron acceptor for oxidative is oxygen. Case of photophosphorylation, it's NADP. And this gets transferred to the Calvin cycle to generate sugar. At the bottom here are some summary statements. Photophosphorylation starts with light. The energy is light and water. And it generates electrons and carbon dioxide and ends up with glucose and oxygen. Now, oxidative phosphorylation is the reverse. It starts glucose and oxygen here. Oxidative phosphorylation starts with glucose and oxygen. Energy is produced and it ends with carbon dioxide and water. And I think, yes, that is the last slide I have. And we are exactly out of time. So remember, well, remember to take the quiz today. And if you have any questions, any more questions or comments, I appreciate the input from you as we went along.
So are you gonna close the test up tonight?
Yeah, it ends at midnight.
Oh, okay. Are we gonna have another quiz before we have another test?
No, the next test will be test 3, and that will be in the last class period in December 8th, Wednesday. No, that's test 3. So the period before that, I'm going to ask you to submit questions about anything you wish. And so the period before that will be devoted to discussing issues of interest to you. And then the next period will be test 3. And the next period will be Wednesday, December 15th. That will be the final exam.
So we will not meet you after the December, not the 8th, the Monday before that, till the last period, last class.
December 8th, you'll have all day to take a test.
Mhm.
December 6th, we'll have a class meeting to discuss questions that you've submitted.
Okay. Thank
you. And the period prior to that will be climate change. And prior to that will be evolution. And prior to that is a week off.
Well, I guess that's it. Happy Thanksgiving. Don't drink and driving. And bye-bye.
Okay, yeah, happy Thanksgiving, everyone, and enjoy the time off. And I'll see you the week after that.
Yeah,
Me too.
bye-bye, everyone.
Thank
Professor, I have a
Thank you.
question. Well, a
Yeah.
thought. So does pH has to do with lactic acid? Like whenever you're running and you're like reach that point of lactic acid, I heard that your pH like gets super down lower. Um, can someone just like eat a lot of like salt prior to knowing they're gonna reach that lactic acid and counteract that effect?
I'm not aware that salt would enter into it. You really should talk to an expert, someone who knows exercise physiology, to know if salt is an important factor in lactate accumulation. I don't know.
Oh, okay. Because I heard some runners taking baking soda before a race, saying that, oh, I don't know, they can withstand lactic acid more because of it. I'm not sure.
Yeah, I'm not sure either. I can see the rationale there that lactic acid is an acid, whereas bicarbonate is a base. And so it would, if you mix the two together in a beaker, they would neutralize. But in a human body, I don't know.
Oh yeah, it's logical. But okay, I'll further investigate. Thank you,
Yeah.
Professor.
Okay,
All right.
take care, everybody. See you later.